Welcome to our deep dive into the fascinating world of amino acids. Today we're focusing on how these building blocks of life are classified. Amino acids are fundamental to biology, serving as the monomers that link together to form proteins. Understanding their classification is key to understanding protein structure and function. Every amino acid shares a basic structure: an amino group, a carboxyl group, a hydrogen atom, and a unique side chain, all attached to a central alpha carbon. The unique side chain, or R group, is the most critical part. It dictates the amino acid's properties, interactions, and ultimately, its role in a protein. Amino acids are primarily classified based on the chemical nature of this R group. This simple division helps predict how an amino acid will behave in a protein environment. The R group's polarity, charge, and size determine its classification, leading to distinct physical and chemical properties. We categorize amino acids into four main groups: nonpolar, polar, acidic, and basic. Each category plays a specific role in protein folding. Let's start with nonpolar amino acids. These are often hydrophobic, meaning they shy away from water. Nonpolar R groups typically contain hydrocarbon chains or rings. They are non-charged and tend to cluster in the interior of folded proteins, away from aqueous environments. Examples of nonpolar amino acids include glycine, alanine, and valine. Their R groups are simple and neutral. Next, we have polar amino acids. These are hydrophilic, meaning they love water. Polar R groups possess functional groups like hydroxyl or amide groups, which allow them to form hydrogen bonds with water. They are typically uncharged but highly interactive. Key examples of polar amino acids are serine, threonine, and cysteine. These are often found on the surface of proteins. Moving on to the charged amino acids, we start with the acidic group. Acidic amino acids contain an extra carboxyl group in their R chain. At physiological pH, this group loses a proton, resulting in a net negative charge. The two main acidic amino acids are aspartic acid and glutamic acid. They are crucial for ionic interactions. Finally, we examine the basic amino acids. Basic amino acids have an extra amino group in their R group. At physiological pH, they typically gain a proton, giving them a net positive charge. Examples include lysine, arginine, and histidine. These positive charges are vital for binding negatively charged molecules, like DNA. To summarize, the classification of amino acids based on their R groups is foundational for understanding how they influence protein structure and function. Remember the four categories: nonpolar, polar, acidic, and basic. This framework simplifies the complexity of the 20 standard amino acids. With this understanding, you are ready to explore how these classifications dictate protein folding and the intricate molecular mechanisms of life.